You're listening to an MRCT Center podcast. We are the multi-regional clinical trial center of Brigham and Women's Hospital in Harvard. Our mission is to engage diverse stakeholders to define emerging issues across all stages of global clinical trials and to create and implement ethical, actionable, and practical solutions. Hello, welcome to our podcast series on promoting global clinical research in children. We will explore various aspects of the multi-layered and complex set of issues related to the conduct of global pediatric clinical trials from a multi-stakeholder perspective. I'm Lisa Koppelman, the Program Director for the Promoting Global Clinical Research in Children program at the MRCT Center. I am pleased to introduce this discussion on understanding platform trials for pediatrics. I am delighted to introduce Dr. Danny Benjamin today. He is the principal investigator and chair of the National Institute of Child Health and Human Development's Pediatric Trials Network. That network is responsible for designing and leading clinical trials of off-patent medicines in children of all ages across all therapeutic areas. Danny has led platform trials enrolling nearly 10,000 participants under an IND in multiple patient populations. Danny is recognized by the National Institutes of Health as a premier mentor and educator, and we are thrilled to welcome him to this podcast today.
Thanks. I'm Danny Benjamin. I'm Distinguished Professor of Pediatrics at Duke University, and I'm chair of the Pediatric Trials Network. The Pediatric Trials Network is sponsored by the National Institute of Child Health and Human Development, which means that it's sponsored or funded by the National Institute of Health. And the NIH is directed by Congress to study all of the older drugs that were developed in the 20th century that were never studied in children. And we're to find out the dosing and the safety and the efficacy of all those drugs in children of all ages in all therapeutic areas. And we've also been tasked to do that with drugs developed in the 21st century that have not been fully studied in children and have lost their patent protection. As distinguished professor, all that means is that I have been doing this long enough to make a meaningful contribution to public health. And one of the things that I've been able to do is figure out how to do platform trials in children. And so I'll talk a little bit about the history and maybe a path moving forward. And I want you to know that we're going to get there on this journey together. We'll get through it together, regardless of our role, whether we're the ones prescribing the medicines or the ones we're not getting the children enrolled in trials, or we didn't do the legislation to require children to be enrolled in trials. We've all been part of not only the problem, but we're also, more importantly, part of the solution. Before we get into the meat of the matter as it relates to platform trials, a few definitions are worth going over because we all read different things in the peer review literature or hear about different things as it relates to research. So master protocols are simply those that can answer more than one question at a time. And they are developed to help improve efficiency of clinical trials so that rather than developing 20 different protocols and funding 20 different studies and getting all of the sites up over and over and over again, we combine all those questions into one trial. And a master protocol might have 2 or 3 or 10 or 20 questions that it's trying to address within it. A platform trial simply means I'm studying multiple treatments or diseases at the same time in the same protocol. So it's the most flexible term. Can be one or many diseases, can be one or many treatments. A basket trial, this is a type of master protocol where one therapy is tested on more than one disease. An umbrella trial is one in which multiple therapies are tested for a single disease. I just want to set the tone for a couple of things and just a particular point in drug development. Okay. That is, it's under an IND. It's Part 11 compliant. That's what we're talking about for those of you who are the Americans, or for under the EMA regulation for the Europeans who are in the crowd. We've got a molecule that has already gotten its first indication. Okay, that's going to be my baseline working assumption. We've already got a first indication in older humans, and we're now taking that molecule into younger humans. Okay, because a lot of times when we talk about platform protocols, we'll talk past each other. Some people are not holding an IND. Some people are doing it for the first indication. Some people are doing it. It's already been already has its first indication. We're doing it in children. That's the situation we're in. Some of them, it's like 3 bros in an Excel spreadsheet in somebody's basement. You think I'm kidding, but that happens. Okay, we're not talking about that. What we're talking about, we've got a molecule. We've done it in adults. We're taking it into children. Okay, just to get us all on the same page. This dates FDA takes its birth date back to 1906, when actually someone had the idea to take horse serum, inject it into children. 13 children died as a result of the fact that it had not really been studied at all before giving it to those children. And so the legislation was written that ultimately provided some protection to adults. But by 1937, a company found a great way to dissolve sulfanilamide. It's a product called diethylene glycol. You might know it by the word antifreeze. And 107 people died, and most of them were children. And this led again to legislation around drugs needed to be safe before marketing them you needed to show that they were safe in adults before marketing them. And then many of you will remember this story, thalidomide. It had been sold in Europe. There was some FDA staff in particular who were really prevented that from being brought here to the United States. And over 10,000 children either died or were born with birth defects as a result of that molecule. And again, legislation was written to protect adults. You had to show that a product worked before you gave it to them. But I want to give you a common theme here. Time and again, children died, and our solution was we wrote legislation for people who vote. We protected adults based on the deaths of children. And so for the first 100 years, over 95% of the molecules that we had available to us, the label says, don't use in children. Not safe for use in children. If you think, gosh, well, we got some studies done, we knew some stuff. Just to give you an example, you might have heard of this molecule. It's called ampicillin. It's given to several 100,000 infants just in the US, humans under 30 days of age. Several 100,000 of them get it every year. For children less than 28 weeks gestational age, when I started my career, children less than 28 days of age, there were no studies for data submitted to FDA on ampicillin in babies less than 28 weeks. There were 6 samples in the entirety of the peer-reviewed literature for people under 28 weeks gestational age for freaking ampicillin. Okay, that's where we were. Okay, for 100 years. BPCA and PREA come along, landmark legislation. By 2006, this is from the exclusivity program, 115 molecules had some data submitted. And there were about 100 key labeling changes, and there were 37 that had substantially different dosing information. And most of those substantial dosing information, most of your surprises come in children less than 2 years of age because of the way drug enzymes develop in humans. Because you get PK surprises. And by PK surprises, I mean like not just
5% difference in dosing, but like 5 times what you would expect to see in dosing. And it turns out that all drugs are toxins, and only some drugs, if you give them at the right dose, help some patients. But if you give 5 times too much, turns out that can be a problem. And if you, for safety, and if you give 5 times too little, you can kill your patient because you're not giving them the right dose. Okay. So the FDA emerges with never extrapolate dosing, and really safety should be assessed on a risk-benefit analysis, but your go-to is not going to be to just extrapolate, but extrapolation will apply to efficacy. So it's true that children are not small adults, but they are also not Martians. That's the good news. Okay, so we can extrapolate some efficacy, and that can be full extrapolation, partial, or none. That's going to be a key question for each of you in the breakout groups. Are we on a full extrapolation, partial extrapolation, or do we have no extrapolation? And I don't want to get into the details of the legislation. There are slides from the days of Diane Murphy, for those of you who worked at FDA 20 years ago, from the days of Diane Murphy, where she and the team at Cedar Peds and OPT really worked out well a lot of this logic. This is like remedial thoughts on extrapolation, just kind of how I keep it in my brain when I'm explaining it to my 17-year-old. So like an example of full extrapolation or no extrapolation, if you have full extrapolation for something like a lot of infections, you can typically do some dosing and safety. If you have partial extrapolation, okay, you can do some, that depends on kind of what age you're at for analgesia and anesthetics. You're going to need to do some masking in your trial, some exposure, safety, exposure, outcome relationships. And then for something like chronic lung disease or BPD in premature infants, if you're using a product like sildenafil, you really don't have anything to extrapolate from. You're going to need to do the well-powered trial or trials. Okay. The topic areas for this workshop are going to differ in their approach to extrapolation, and I'm not going to give any advice about that from here. Okay. By the way, divisions can change their approach to extrapolation. Samadi is here, who was amazing when she was for anti-infectives, and we would go back and forth and talk about do we really extrapolate here or not. So I'll get in, by the end of the talk, I'll get to really what the 3 stages of research are. But I was in the first stage of research for platform trials. The Stampede trial had just started enrolling, and now over 10,000 people have been enrolled in that. But most people, myself included, was not aware of the Stampede trial. And I was rounding in the NICU, and I saw once again a baby with complicated meningitis. And we didn't know the dosing, and we kind of guessed. And I said to the bedside neonatologist, Brian Smith, I was like, I'm tired of guessing about this dosing thing. We got to do something about it. And I said to him, I said, if we just get a sample from every baby and we tell the moms you're going to get this drug anyway, but just give us 1 drop of your baby's blood and we'll bank it. And then we'll get other babies who are getting the same drug. And eventually we'll be able to describe the dose. Okay. And so we proposed that, and people thought we were delusional. And we were told it would never work. But that's where we were. We were at stage 1 of research. No one can do this. It's impossible, even though people were doing it with the Stampede trial. No one can do this. It's impossible. So Brian and I started moonlighting. And in those days, you could moonlight, and you could take the money from moonlight and you could put it into a discretionary account and do research on it. So we'd moonlight on nights and weekends, and we'd take the money, and we started doing this little opportunistic study. And we started with antifungals because that just happened to be my area of interest. And we had gotten a little bit of data from a few antifungals. These have been available at this time for about 4 years. not micafungin, caspofungin. Voriconazole had been made available in 2002. And we had some data from micafungin, which was a little bit older of a drug, maybe 2 years older rather than 4. And I got a phone call about this baby from another institution, and it's a 650 gram boy, 24 weeker. And these lesions here are on his back, and they had grown while he was getting 7 milligrams per kilogram of lipid complex, and they were enlarging on therapeutics. And they were like, "Hey, man, we're planning on withdrawing life support, but we heard you were doing some work on some of these new antifungals." And I said, "Well, based on what the companies are doing and what you're seeing in the peer review literature, they're off by about tenfold in their dosing. It's not the fault of the company. They just done the first study, then, whatever. It's off by what the models expected us to see. And so change the dosing of the echinocandin and the voriconazole. Don't withdraw life support. Call me tomorrow." Okay, and we'll keep talking about it. We'll talk about how to get levels. Okay, sometimes things work out, sometimes things don't work out. I'll get to the end of this story by the end of the talk. But at this moment, right after this phone call, which is about 11 o'clock at night, I called up Brian at home, and I was like, "We're doing the right thing with this opportunistic study, and we got to do it for every molecule that's used as an anti-infective in the nursery." And he was like, "All right, I'm in." And I called up a couple of partners at the Pediatric Pharmacology Research Unit, which is an NICHD network, and I told them the same thing. I said, I'm going to do this for my career, and I'm going to describe every anti-infective of meaningful use in the nursery during my career. And several of these people were my friends, and they laughed out loud. They laughed out loud. They're like, "There's no way. There's no way this can get done." I want to give you an update on my career goals. These are the first 150 molecules that we've studied. My grandfather worked until his death at age 95. So did my grandmother. And my mom and dad are still working. So I'm not done, but I'm on my way. It's 100, and that we have described some aspect of something about the molecule in some patient population. Okay, so for ampicillin, it's babies less than 28 weeks gestational age. For acyclovir, it's premature. For some of these molecules, it's children with obesity, and others, it's children who are on ECMO. It turns out actually for extracorporeal membrane oxygenation, if you take all the blood out of a human being and you run it in a machine round and round and you oxygenate it and then you cram it back into the human being, it turns out sometimes dosing's different. I know it comes as a surprise, doesn't it? It comes as a surprise. It turns out we've done more of that work than all other research groups in the world combined. It just hadn't been done. We learned a few things along the way. These are your tax dollars, by the way, if you're an American. Thank you for paying your taxes. And you say to yourself, "Well, gosh, are you just doing these studies? Or are you doing it for labeling?" Yes, we're holding an IND. Here are the IND numbers. This is about an 8-year period here where we've taken molecules all the way through from the platform trials all the way to labeling changes. Sometimes the CSR, that's the clinical study report, we're negotiating with the agency right now on what things should say. We've either completed enrollment. This is just kind of the last, this is 2018 to 2024 years worth of work in the program. And what happens is the molecules go into the opportunistic study first. Then we make an assessment with FDA as to whether or not we just need to do dosing, or if we need to do dosing and safety, or we need to do dosing, safety, and efficacy. But we get them into the platform first, okay? And we've got Just for fun, we started doing breastfeeding studies. We did the obesity related research, because it turns out obesity, just those numbers have really changed in the last 30 or 40 years when all these molecules were developed. Do you actually get labeling changes? And yes, this is about a dozen of them that have used one or more of the platform trials. Breast milk study, the agency came out with this guidance on breast milk. Good for you guys, by the way. They came out with guidance on doing these breast milk studies, and it got released. And I looked at the guidance, and we have this area outside of my office. There's some smaller offices across the way where the junior faculty sit. And then there's between us some open area cubicles. And every once in a while, I'll read something and I'll come out of my office. And for those of you who have ever been to SeaWorld, all the junior faculty call that area the splash zone, which I loved until I realized that they were comparing me to Shamu. So I came out of my office. I was like, oh my gosh, there's this new breastfeeding guidance. We're going to be the first people to do this before anybody else figures it out. So turns out we've gotten our first labeling changes. These are the molecules under active study, and we've got the machine going for breast milk. Turns out there's some stuff there that I didn't expect there either, just like with ECMO, just like with obesity. So some overarching lessons learned. We use the opportunistic study as a funnel. So we have this opportunistic study where we put in all the molecules and we figure out, we screen investigators, like how often do people enroll? We screen sites, over 300 sites have enrolled in at least one PTN trial. We screen molecules. We put 145 molecules into the platform. We screen populations. Can you actually get this done during safety or efficacy? And then we also do emerging and difficult to get to populations. So we have over 2,000 samples from patients who are on extracorporeal membrane oxygenation, 30 some odd thousand biologic samples, over 100 validated assays. Right now, 50 assays are moving through the FDA pipeline. And then we take the molecules where we're seeing success in enrollment, and we put them into other trials, some of which are platform. For example, SCAMP is an example of this. SCAMP is a nice example of where you can do, it has an example of both partial extrapolation and full extrapolation. When we started, we were randomizing the infants. We do randomization and PK/PD with masking. And then we changed over to simply doing the last groups of infants were not randomized. We did this with safety, with antipsychotic medications. These are three of our more recent studies, and they're nice examples if you are in either, I need some safety, I need to do full or partial extrapolation, or I need to do a pivotal study. This pivotal study was actually in adults. It enrolled over 1,900 adults. Abatacept and infliximab were obviously licensed. Cenicriviroc had not been licensed yet. And the nice thing is that because these protocols were paid for with your taxpayer dollars, you're welcome to them. And turns out they're freely available. Okay. And so some other detailed lessons, it really is, if you can make it so that you've got multiple molecules going at the same time, really helps the sites out, because they're just trying to make ends meet with their study coordinators. It really helps to work closely with your key stakeholders. So for industry, this is obviously different sometimes than it is for an NIH funded study. You've got to deal with multiple different continents. We've only had to do that a few times. We're really getting to regulatory grade development of this stuff. The lab stuff can be really tricky because you're looking at microsampling of these molecules, and working across matrices with breast milk can be a real hassle. Not that breastfeeding is a hassle. I'm not allowed to say that as a pediatrician. I actually learned quite a bit. All four of our children are adopted, and so it's been the breast milk study has been really interesting. When you're thinking about a platform study, really flexibility, okay? Because you're typically going to be working multiple different companies. Like if it's one company and three different molecules, that's a layup compared to 3 different companies doing this. Okay. And let me tell you, there's going to be some give and take. In exchange for the efficiency of the platform, you're going to be giving up a lot of, I'm doing it this way because I'm just talking to myself. I've talked to myself and I agree with myself again. You don't get to do that in a platform trial when you're working with multiple companies. Okay. One of the things that can be a real problem is a lot of the platform studies is kind of a choose your own adventure. And we found this with Active 6. Active 6 studied ivermectin, which a group of people thought would be very effective to give to people with SARS-CoV-2. We had the radical idea for hydroxychloroquine and ivermectin that if you gave the patient enough hydroxychloroquine to kill COVID, you would also give them enough to kill the patient. And it turns out that's correct. But people were very, very interested in enrolling in the ivermectin arm. Okay. And I mean, there was definitely equipoise. People believed both sides very strongly. It's an ethical study to do. That arm enrolled incredibly quickly. Okay. And if you're on a choose your own adventure, people may not be choosing your molecule. Okay. And so you just are kind of going to need to work through that through a diversity of sites. One of the nice thing, however, is if you're doing a diversity of sites to get your enrollment done for your molecules, you're often, not always, having enough diversity in sites that you have diversity in the humans who are receiving your product, which is an unmet need right now in most studies. You really want to align the goals of the study with the study setting. So when we first started going into the antipsychotic medicines, we had really done a lot with inpatient studies, but we weren't as strongly connected to outpatient investigators. And so we were oversampling young children because we had so many inpatient investigators. And of course, we were seeing surprises based on the metabolism of those molecules because of the age of the young children that we were seeing. And so we had to pivot in a lot of our enrollment strategies midstream. And just you're going to see some of the surprises in these trials when you do this. And to have the ability to pivot midstream is really important. As most of you all know, there's 15 or 20 clinical divisions within a typical department of pediatrics. And so when you're talking about, Duke or UT Southwestern or Harvard or whatever as a site, that's not one site. That's like 20 different sites at best. Because even within each division, somebody's like, oh, I'm the person who does BPD. Oh, I'm the person, who does blood pressure medicine in the NICU. And it's really the investigator and the study coordinator. And so you can get really variable performance. The, EHR to EDC, electronic health record, to the electronic data capture. We're at stage 2. I'll get into the stages of research later. We're in stage 2 with EHR to EDC. But you really, when you're talking about some of the things about when product was given, you can run into real problems with the electronic health record. Oh, I want to give you some follow-up. This baby, it turns out the dosing that we recommended was based on the sampling that we'd done was correct. And this baby went out of the NICU in a car seat, has hearing intact, vision's intact. The cognition scores from the Bailey score for this youngster can be mainstreamed in school. I just want to say a few things about that. If you're this baby's mama, the products were available. But because we didn't know the dosing, we were going to take her boy off of life support. And because we put the molecules into a platform trial and we got the work done early, this boy's in high school. So you're getting ready to embark on doing platform trials. And you may wonder if it's worth it. And it's my experience that it is. That it often does not feel like it's worth it to save a few months or a few years to get the information out into the public domain, but sometimes it is.
Ultimately, my hope for all of us and for children's research is that we're able to improve the efficiency of studies in children. These are research questions and answers to these questions that seem routine 5 or 10 years from now. Because what seems impossible today is often not only possible, but routine. Thanks.
Since 2007, the MRCT Center has been an independent, pre-competitive convener engaging diverse stakeholders from industry, academia, patients and patient advocacy groups, nonprofit organizations, and regulatory agencies to deliver ethical, actionable, and practical solutions for the global clinical trial enterprise. Our resources are freely available at mrctcenter.org. Follow us on LinkedIn and sign up to receive our monthly newsletter on our website. Finally, please like and subscribe to this podcast channel. We look forward to many exciting discussions to come. Thank you for listening.